back for Science in Pajamas. So today we're going to talk about a little bit of the history of modern genetics. So for long time, people knew that something was being passed on from parents to offspring, that traits and all that were being passed on from parents to offspring. They didn't really understand how that worked or what was doing it or anything like that. Hi, Bradley. But they did understand that traits were being passed on. Now, let's jump to the 1800s. In the 1800s, in what is now called the Czech Republic, it wasn't called that then, but we have a man born whose name we know as Gregor Mendel. Or as my kids hear me say, my man Mendel. And he's very important to the idea of genetics because he's actually what we consider the father of modern genetics. So he was born in what's now the Czech Republic in the 1800s. He was born in a very agricultural community, so not a whole lot of money, very in the earth type people. And he actually went to go study at the University of Vienna. He studied mathematics, physics, botany, anatomy, and eventually he ended up as a monk in an Augustinian monastery. And it was there that he started his experiments on genetics. Now, he worked in the monastery garden, and there he noticed that there were some pea plants, just, your standard garden variety peas. But what he noticed is that they kind of came in a few different traits. So he noticed that the tall ones always had tall offsprings, and the short ones always gave rise to short offsprings. The ones with purple flowers always gave rise to offspring with purple flowers. The ones with white flowers always gave rise to the ones with white flowers, and so on, so on. Now, we call this true breeding because we know exactly what's going to be present in the next generation. And the reason why these were considered true breeding is because they were self-fertilizing. So one plant would use its own flowers to fertilize its own flowers, and so it kind of kept the genes pretty much the same each generation. He's like, well, I wonder what would happen if I was to take some of the pollen from this one and mix it with the pollen from that one. So what he started doing was, these are called hybridization experiments. And that meant that he was kind of looking at different methods and different ways to fertilize them. So let's say we have a tall pea plant. So, and then he decided to cross it with a short pea plant. Now, what he thought would happen when he took the pollen from one and used it to fertilize the other, what he thought would happen is, okay, we got tall and we got short, therefore the offspring will be medium. But you want to know what he found out? All of the offspring were still tall. So every single offspring that arose from that cross was tall, just like one of the parents. None of them were short. None of them were medium. Like, all right. Well, that's weird. We have tall plus short, and we get all tall. So this was the original parent generation, and this was the first offspring generation. Now, today we call that the F1, the first filio generation. So he's like, all right, well, what would happen if I took the offspring, the first filial generation, and I crossed them together? So he would take the pollen from one and use it to fertilize another. And what he got that time, you ready for this? Let me get my ground first. Still stay on the screen yet? Yep. And what he got was tall, 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 short. So what he noticed is that when he crossed the offspring of the initial one, so when he crossed this F1 generation, he got roughly 3/4 or 75% were still tall. And about 25% were short. So that led him to doing other similar experiments with, for instance, flower color. Okay, well, what happens if we cross a purple one with a white one? They got all purple flowers. Cross that generation of all purple flowers, and you notice you got about 75% purple flowers in the F2 generation, the second filial, the second offspring generation, and about 25% white flowers. Did the same thing with pea pod or pea color, pea pod texture, and he noticed this consistent ratio. So that's what led him to a couple of the other such, later on led to the laws of segregation, laws of independent assortment. But this study and his analysis of it is what led to genetics that we study today. And that's why Mendel's called the father of modern genetics. Now, is this the end-all be-all pea plants? No, it's not. But from there, from what he studied and what he came up with, and then additional studies after that, like I said, that's what led us to our idea of genetics today and how we still continue to understand and teach it today. So what we know about genetics today is that, first of all, it was a good thing Mendel started with the pea plants because in terms of their genetics, they're a lot less complicated than humans. Also, very quick generations. But apart from that, we were able to come up with some terms and be able to define them. So let's start off with a few basic terms. We have genotype and phenotype. Very similar words, not the same thing. So what genotype is, that is the actual allele description, the description of the genes. So that is the genetic representation. Genetic representation. I ran out of space, I'm sorry. So genotype refers to the genes. So far, so good? Phenotype is the physical description. So the physical representation. Blah, 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 blah. So phenotype is the physical expression. In other words, what do we see? What is the actual expression of the genes? Let's go back to Mendel's pea plants for a moment. So there is a gene for height in pea plants. And we know that there are two variations, tall and short. Now, genes can be thousands, tens of thousands, hundreds of thousands of bases long. We do not want to write out all those bases. Kids don't want to write it. I don't want to grade that. So what we agree on are these things called alleles. Now, alleles are variations of genes. Like we said, there's one gene for plant height in the pea plants, but there's two variations, tall and short. Those are the alleles. So you have alleles that code for different expressions of the same gene. And what we kind of have this agreed upon system is to use letter representations for the different alleles. So since we saw tall more commonly, let's use the letter T. Now, as you remember, just like with us, pea plants, their genes come in pairs. Our genes come in pairs because our chromosomes come in pairs because we get one from mom, one from dad. It's the same way with the pea plants. So how that works is you should have 2-letter representation for the genes of that particular trait. So if our trait is height, the height of the pea plants, we would use 2 letters to represent it. And that's, don't worry, I'm going to tie it back up with here. So we know that there are 2 expressions. We have tall pea plants and we have short pea plants. Now again, we like to use 2-letter representations for the genes, 1 letter per allele. And again, you have 2 alleles because you have 2 chromosomes. And how that works is, most of the time, it doesn't really matter what letter you use as long as you're consistent. And there are some where the letters are kind of already assigned, but let's just say for this one, we saw tall more often, so I'm going to use the letter T. So to be a tall plant, now we have 2 alleles. That means I have to write 2 letter T's. Now, I'm going to keep it using only T, but I have to differentiate between tall and short. So maybe I can put a capital T for tall, since we saw that one more often, and a lower case T for short, since I saw that less often. Now, yes, I know some of you are thinking, but don't you mean dominant and recessive? Don't worry, we're going to get to that. So since there are 2 variations of the letter T, let's look at the combinations. I can have big T, I can have big T, or I can have little t. Each one of these is a 2-letter representation of what the alleles mean, or of what they are in the actual genetic code. So these are your genotypes. They are the genetic representation. So this represents one allele for the gene, this represents the other allele for the gene, and so on, so on. So these are your genotypes. See, genes, genotype. What the actual physical expression, what we see is tall or short. That's your phenotype, physical. So what do these mean? They mean these expressions. So the genotype is the genetic representation, and the phenotype is the actual expression. What do we see? What is different in the organism? So far, so good? All right. So that brings us to a couple more vocab terms. Now, as we saw, we can have the variations be big T, big T, big T, little t, little t, little t. We can further describe these, and we're going to use a couple new terms. We have, first of all, homozygous. If you remember from our sexual reproduction video, we mentioned something called zygotes. That's the fertilized egg. So that's kind of what this portion refers to. Zygous, zygote. Homo means same. So what we're saying is, essentially, we have 2 of the same alleles that are present in this zygote, which will then become the organism. So 2 of the exact same alleles. Now, where do we see 2 of the exact same letters up here? If you answered big T, and little t, you would be correct. And we would call this being homozygous dominant and homozygous recessive. We'll talk about the difference between homozygous dominant, about dominant and recessive in a little bit. Now, there's another term, because if you notice, we still have one more variation. We call this heterozygous. And again, what that means, referring back to the zygote, so when after fertilization, when the genes come together. And then hetero meaning different. So we have different alleles for a gene in our zygote, in our organism. So where do we see different versions of the letter T? There we go. That's heterozygous. We don't even have to say heterozygous dominant or heterozygous recessive because of the nature of what dominant and recessive means, which we are going to talk about in just a moment. So homozygous, the same versions of the letter, so two of the same. Heterozygous, different versions, different alleles. All right, so now let's get to dominant and recessive. Now remember how in the parent generation, we started out with a tall plant and we crossed it with a short plant. And then in the F1 generation, we got all tall. What that tells us is that the allele for tall height in pea plants is stronger than the allele for short. And that's where the idea of dominant and recessive comes in. What that refers to is variations or alleles for a gene that are more likely to be seen and that will actually override another allele. So what we mean by that is we start off with, going back to the parent generation, let's use our genotypes. So true breeding tall would be homozygous tall, so big T, and then we cross it with a true breeding short, which would be homozygous short, so cross it with little t, and all of the offspring in our F1 generation, and we'll get to this when we talk about Punnett squares. How this works is you get one allele from each parent. This parent can only give the big T allele. This parent can only give little t. So we end up with big T, little t. Now these plants, they are heterozygous because they have two different versions of the T allele. However, we saw every single one of those plants was tall. So what that tells us is that this capital T, the allele for tallness in pea plants, is stronger and will override the recessive short T. And what that means is it will always be seen. Its phenotype will always be tall. Now, just because it's expressing the tall allele and hiding, not expressing the short allele, doesn't mean that short allele isn't important. And the reason for that being is, like we saw with the picture a while back, if we were to cross two of these from this generation, so big T, little t crossed with big T, little t, so two heterozygous plants, and we get to our F2 generation, now we're going to see something different. Now, again, we'll talk more about how the alleles separate and we can figure out the offspring via Punnett squares in another video. But for this one, like we said, we'll just kind of do it. One allele from each parent. We can have big T. That's the genotype. The phenotype would be tall again. This big T with that little t. Still have the dominant allele, so he's going to be tall. This little t with that big T, but we always put the capital letter first. So big T, little t, also tall. Or we can have two little t's come together. So that means we have a possibility for a short plant. And this is the ratio that Mendel saw. 1 to 3 out of 4, or 75% chance of being tall offspring, and 1 out of 4, or 25% chance of being short offspring. So again, when we talk about dominant or recessive, we're talking about the allele that is more likely to be seen. So the one that can actually override the other. We always write dominant alleles as a capital letter. So in this case, the capital T represents the dominant allele, which would lead to a height of tallness. And then the lowercase t is recessive. It leads to the trait that is least likely to be seen. So the only way to show a recessive trait is to have 2 recessive alleles, because now you don't have any type of dominant allele hiding its expression. So when you have 2 recessive alleles, that's the only way you get to see a recessive trait. If you have 2 dominant alleles, or a dominant and recessive, if you're heterozygous, the presence of the dominant one will always win out. It actually overrides the other alleles. So that's it for our introduction. Yes, hi baby. That's it for our introduction on genetics. I hope this helps. We will be doing videos on Punnett squares and all that good stuff coming up soon. But in the meantime, stay well, stay healthy, my friends, and enjoy the science.